Hey y'all, happy Monday. Today is Monday the 20th. Y'all, it's one more week of school and then finals and graduation, which is, to be honest, a little bit lamer than usual. Graduation is a rite of passage that I'm quite sorry that you guys will miss for those of you graduating. Hopefully we will be able to celebrate in like a physically close space, like not that close, but close space, sometime not too far from now. So hopefully you guys saw, I sent you a Canvas announcement a little while ago. I'd like to tell you, I wish it was going to be, I don't want to say a better announcement, but something different. Hold on, I want to pull that back up so I can make sure that I tell you the exact same things I told you like 4 hours ago. So a couple of things. One, today we have class. On Wednesday, we will have a slight class. So today we'll talk about techniques, kind of along the lines of how does all of this fit together? Why would you want to do this? What can you learn? How would you do this? So that'll be the last one of these. On Wednesday, it will be a review. If you have a question that isn't, will this be on the final? If there's a topic where you would like a little bit more information, I will make a general overview of slides and then kind of talk through those. I will also have access to the board, kind of do that. So today, if it will be molecular techniques, techniques review would be if I was taking notes today. That's what I read at the top of the page. So we'll kind of talk about how we think about it from a global perspective. And if you have any questions, I'll answer those. So I'll do that on Wednesday. And hopefully it will go, I don't say well, but we'll talk about that on Wednesday. If you have a question, so I know after the figure analysis 1, some of you commented that you watched this later. If that is you and you have a question and you'd like me to address that, please send me an email. I'm happy to include yours in the comments and the questions. I will not say who you are. If you prefer to lurk and never read a comment, you can also send me an email there. So next, finals. Next week is our final. Our final was scheduled for April 29th at 3 p.m. So the final is going to be a take-home test. So we have done take-home tests for everything else. So we're going to do the same thing. It will be the entirety of the exam. So it will be a full 200 points take-home. I think all of your homework, I'm missing at least one homework, which is on tap for today to be graded. But what you can see is you should have gotten a score. If you were to open your homework assignment, you can see where I wrote on it in red. Oh, sorry, my accent's heavy today. I wrote on it on my iPad, so you should be able to get your score that way. The exam, I will post it on Friday by 5. From then, you have until Wednesday at 4 in order to submit it. Wednesday at 4 p.m., it must be submitted on Canvas. No late work is accepted. If you have if that doesn't work for you, please reach out. Please talk to me. We will come up with a solution. We will make it work. So the usual collaboration rules apply. So you and only you can work on it, but you can use your notes. You can use these YouTube videos, and you can use the internet. So it is open book, open notes, just not open people. Last minute questions, you can pop into the comment box. So I was talking about office hours. So if you have questions, you can pop into the comment box. I have office hours in the usual time slots that I've been having them all semester. So you can come into any of those with questions. You can access the Zoom link from the homepage. It takes you into a meeting. I'll be there. Next week, because your final is on Wednesday, I am having office hours from 1 to 2:30 on both Monday and Tuesday for you to ask questions. There won't be any on Wednesday, mostly for the reason that, like, at that point, you've probably already gotten into anything you need. If you cannot make either of those times, I know a bunch of you guys are working and watch these late at night. You can still, like, I am happy to do that. Tina asks, can you write it on regular paper? Yes. Thank you. I meant to say that. So if you don't have a printer, because like I don't have a printer, you can write it on regular paper. You do not have to write the questions. You can just write like number 1, then like somehow indicate 1, write your answer, and then move on. If you use regular paper, I will always try to write like 3 out of 5 so that you can tell where the points have been lost, kind of the same way it would be on anything else. But you can do that. You can upload it on Cam Scanner. So in any way, shape, form, or fashion. If you would prefer it to come in a different format, if you don't have a printer or an iPad or some other way to do that, I can send it to you as a more streamlined Word doc so that you don't have to scroll through lots of pages. If you'd like that, send me an email. Something else about the exam. I don't know. When I think of things, I'll tell you. The other thing to note that was also in the announcement today. So if you look at the original syllabus, like the OG syllabus from January, which to me feels like 10 years ago, probably to you too. Sorry, I do not have the COVID. I haven't left my house in 5 days. I'm dying. So the total points for the course is going to be 670 instead of 700. I realized that it was not pertinent. It was kind of silly for me to try to find another way to give you 30 points. So what this means is that the point scale that tells you, okay, if you get this number of points to this number of points, you get an A. That means nothing. What is important is if you get 85% of the possible points, which is 570, all the way up to 670. I think that's right. That doesn't feel right, but math. Then you get an A. So on Canvas, on your grade page, it gives you a letter grade. That letter grade is correct. The point value, because I do run a points-based course, is no longer correct. I will post that in a later announcement this week, but I was trying to give you guys as much information as possible. So I'd like to apologize, but I feel like everyone is relieved to have one less assignment to do. So it will be a 200-point final. We will just have slightly less points for the semester. In case you are late or it glitched, there is no timed portion for the final. It will all be open notes, open book. You'll have 5 days. If you need more time, please send me an email. If you have not necessarily other finals, but if you have a job or you are a caretaker for your family or you become ill, please reach out. I want you to do the best that you can. Anyone have any questions about the announcements that I sent you on Canvas or about anything else? So today, so molecular techniques or biochemical techniques or biochemical assays are really valuable. So when we think about techniques, so as a biochemist, I think about being able to answer this question: what does this protein do? Right? So we know that today there's a pandemic. And so today there's a lot of information about I think I saw today that there are 29 proteins that COVID-19 makes, just 29. In your body, you probably have thousands upon thousands of different proteins. Bacteria have, somewhere on the order of 6,000 genes, and so those proteins do jobs. So when we encounter an unknown protein, we want to be able to ask, what does this protein do, or how does it do it? And so all of these techniques are designed to be able to do that. Earlier in the semester, we talked about FRET, right? And so FRET is going to allow you to determine, how does this protein open and close? Or do these two proteins pass? Do they come together? Or are they just hanging out in not the same place? So there's that information. When we think about other types of info, it's worth considering, how does everything else work? And so one of the things that I think is really important for us to kind of consider is, why do we want to do this? How do we go about doing this? And why does it matter? So on Wednesday of last week, we started talking about bioinformatics, and I forgot to post those handouts. Shoot. Hold on. Let me write that down, because you might actually care. And I just have the end of the semester brain. Okay, now I will maybe remember after this class to post the old bioinformatics handouts. So one of the things that I think is really important and really creative, and one of the ways that biochemists are different than other types of chemists, is we want to use all of our resources to ask a question. We have a protein in a new organism. What does it do? So if you were to think about that, there's kind of an approach here. So let me move my chair. Hold on. So today what we want to think about is, what do we do if we discover a new protein? So the question is, we have a new protein, or maybe we have an organism that we found this thing and we want to know what it does. So the first thing I would likely do if I were to, we have a protein. Now, this protein was discovered either based on the gene, we have the sequence. In order for this process to work, so we're going to kind of think about these as caveats, we have the sequence. So whether we found it in the genome, like we sequenced the genome and learned about it that way, or in other ways. So once we have the sequence, we can, one, use BLAST / other bioinformatics tools, to predict function. So BLAST sequences, we can blast a protein. If you have an unknown protein, you can put it in, and once you hit go, it will pull out things that are similar to it. So the advantage of that is, this protein in your organism, it's the only one of its kind, but we can use these tools to predict the function. So whether we think sequence drives function or function drives sequence, maybe we can guess what our protein does. And so if we can predict a function, we can design a set of experiments in order to do that. So bioinformatics could also give you information about sequence. It could give you information about hydropathy, how hydrophobic, stuff like that. So BLAST and bioinformatic tools can predict function. There are structural predictors. The ones I have used don't always work. You need a lot of information, and you should know that it's not the best. You can do it. Sometimes it's really cool and works, and sometimes it just kind of, these days it might be a dumpster fire, for lack of a better way to say it. You could fit it into that model, and that model might not work. So, but there are structural predictors as well. So we can use BLAST and other bioinformatic tools to predict the function. So two, if this was me, I'd try to figure out, what does it do? And then I'd want to subclone it. So subclone for use in experiments. And so the other thing we could also do is use RT-PCR or other techniques to determine the mRNA concentration. So RT-PCR in this case would be really advantageous because, so we could say that the mRNA concentration is there. If your protein is upregulated during certain environments or downregulated or constant, that all of that information tells you things. So one thing that is often different about biochemical or biophysical experiments is that all information is useful. So you could subclone for use in experiments, and we'll go through that again here in just a second. But if you want to ask, like, is this a methyltransferase? Yes is one answer, and no is a good answer. So the trick with biochemical experiments is that the answer can be no. Now, no is annoying because you have to show that the answer is no in more than one way. Because it could be that you didn't ask the right question, you asked the right question in the wrong way, or you have a persnickety enzyme. So no is often complicated, but yes has its own issues. So let's talk about what subcloning means. Also, as always, if you have questions, feel free to pop in. Otherwise, we will continue to chat. So, subcloning. This is a term that is kind of a general term, and it basically means to create a plasmid, so that's a DNA construct, for heterologous expression. So heterologous expression, this means expressed in a different organism. Or expressed from a plasmid. So when we think about heterologous expression, so heterologous expression can mean a couple of things. One, we're going to take in this case, we'll talk about a human protein, and we're going to express it in bacteria. So it is different host. We could also express an E. coli protein from a plasmid in an E. coli. So the heterologous usually do it not. But the other thing that's worth thinking about is that it's not just, so you can express an E. coli protein in E. coli, but as soon as we modify the promoter or use an IPTG promoter, both of those are going to tell us that it's heterologously expressed. So to subclone, we want to create this DNA plasmid, and we'll review what a plasmid is here in a second, so that we can express the protein. If we can express the protein, sorry, standing in front of the board today. If we can express the protein, then we can start to ask questions. What does this protein do? Because our goal is to understand what it does or how it does it. So if we think back to vectors, vectors are these circular DNA pieces. So these are circular DNA that control protein expression. So if we remember so heterologous expression is the ability to create. So Ryan asks, could you repeat what I said? So heterologous expression is the ability protein and express it in fact in E. coli. So that feels like it should be homologous expression. So heterologous, those types of things, it's going to be heterologous expression. So heterologous expression allows us to create a protein, and now we can ask, what does this protein do in a variety of experiments? Good question. So we have this circular DNA that can control protein expression. So the first thing we want to think about is we have our vector. And in our vector over here, we are going to have resistance. This resistance cassette will encode for an antibiotic protein or a protein that degrades an antibiotic. So these can do a variety of things, but it is a selection marker to allow us to determine when our protein is expressed and how it functions. We are then going to have a promoter. Before we encounter the MCS, right? So the MCS is the multiple cloning site. So in the MCS, So the multiple cloning site is going to allow us to find a variety of restriction enzymes. Those restriction enzymes are going to allow us to insert the gene. And so we're going to have our same plasmid. And this is not to scale. So we're going to have a promoter. Same promoter. Now our multiple cloning site is going to be chapped. And so when we now have separate ends. And we'll see if this marker works, and if it doesn't, we'll pick a different color. Blah, blah. I think this one works. That'll work. So this is our gene. And that gene was likely amplified using PCR out of the genomic DNA. And so once we amplified that, and down here in the end, we have our resistance cassette. So once we have a resistance cassette, what we're attempting to have here allows us to selectively decide when we are going to express this protein. We can add IPTG. There are a few other sugar molecules you could add. So then we're going to put this using transformation into a host system. So once we have a vector, we've created this. We are going to confirm that we have that using sequencing. So we'll talk briefly about sequencing, then we'll talk about transformation, then we'll talk about sequencing. So transformations can be electrochemical, meaning we use, or we'll put it in quotes, shock to force DNA across a membrane. So basically, we're going to use the natural charged nature of DNA and shove it across the membrane. Once you put it inside the cell, we can start to use it. But DNA next to cell does not a protein make. So we can use electrochemical. We can also use heat. And so this basically uses, you could swap out the word shock for temperature. So you could use heat. You could also use chemicals. So this is a little different. So for a chemical transformation, we're going to create small pores, otherwise known as holes, in the membrane that DNA diffuses through. So DNA has to get across the membrane. We can do it in one of these three methods. Heat shock also creates pores. Heat is a little bit different. You basically go from hot to cold, and the cells basically do this, and then the DNA gets inside. So in this case, the transformation is how we can get the DNA across. So now we have an E. coli cell, or a insect cell, or a plant cell, or some other type of cell with DNA on the inside. Once we find DNA on the inside, we can then start to ask questions about the DNA. This is all before we express protein. So it feels like, oh, it's so easy to create a heterologous construct. We're going to just make it happen. So the thing with this, and the way that we really want to think about it, is we got it across the membrane, but we'd like to be able to confirm that we have the protein we care about, right? So you can do that using restriction enzyme digest, where we can look at the DNA kind of on a qualitative level. So we'll say, actually, I know that it lags, so if I erase that, someone will ask about it. So transformations are the method by which DNA gets across the membrane, and it has to get inside in order for it to be useful. So the next thing we want to do is we want to analyze what we have inside the cell. So we're going to use DNA analysis. So this is to ask, did we create the construct we wanted? So how does the DNA just come out? You have both, so diffusion plays a role here. If you use electrical charge, when the electrical charge oh, okay. Sorry, if more than one of you said it was frozen, then I was going to try to figure it out. I'm glad it worked. Hopefully it's better now. It looks like it's about to rain, so I'm sure that's going to just make everything terrible. So the question was, well, I'm sorry it paused and freaked out in the middle of the answer. So, all right, let's go back to the beginning. It doesn't tell me that it's being weird. So Maggie, if it's weird in the next 5 minutes, will you let me know? I mean, there's not a lot I can do about it, but I can always end this stream and start again. So if it's still misbehaving here in a second, let me know, and I'll basically just end this, start again. Because that's, I feel like how life works, right? Just restart. So the answer to Johnny's question that was asked several minutes ago so the question is, how does the DNA go through the pore? Or Tina says, refresh, change the pixel count to the highest one, and it will go clear. Crowdsourcing. So in this case, we have our supercoiled DNA, so it's quite small. And in this case, the pore is open. Now diffusion, because we have a higher concentration of DNA on the outside than on the inside, will force some of the DNA in. So we see that. If we use the electroshock, where it basically charges it and pushes it across, that will force it across the membrane based on the electrical charge between the two plates. So the other thing that's worth thinking about is, how do we get it across? If you're using heat shock or chemical shock, where you basically degrade the membranes and get some to go in, the other thing that we do is you add a lot of DNA. And so this process for transformations is relatively lethal. Basically, it allows some of it to go across, but not all of it. And you run the risk that the cell dies and all the insides go to the outside and everything else. So transformations, you want to have a lot of DNA or a lot of cells, likely both. And so when you do that, it allows the DNA to go across. Are there other questions? Were there any other questions about transformations? Otherwise, we will go back to DNA analysis of the plasmids inside the cell. So DNA analysis. Did we create the construct that we wanted? So there are two different ways that we can kind of do this. And so likely you would do both. Option number one, or more like avenue one. So avenue one would be a restriction digest. So one of the things that I like about restriction digest is it's pretty fast. We have DNA. You can cut it and get a qualitative answer as to whether or not it's correct. Hold on. You can go out. Sorry, it's 5:05. The dog thinks it's time for something. Anyway, I don't know where she thinks she's gonna go. But so the question is, a restriction digest, this is fast. And I would say it's qualitative. So if you inserted a gene, you can see that basically, hey, the DNA is roughly the right size. This doesn't tell you, is everything about what I put into the cell correct? So then you would have to use sequencing. So a sequencing is going to give you a quantitative results. You will get an exact DNA sequence. So this will give you a sequence of DNA for your construct. So it is qualitative. No, it's quantitative, meaning it will tell you exactly what the DNA is. Be right back. I'm going to restart this. Stay here.